This video will cover the essential topics of proteins and amino acids, the building blocks of life. Proteins are fundamental molecules that provide structure, catalyze cellular reactions, and carry out other vital tasks. They have multiple biological functions and are classified based on their specific biological roles. Enzymes are a varied and specialized type of protein with catalytic activity. They are essential because they catalyze all chemical reactions involving organic biomolecules within cells. Transport proteins bind and carry molecules or ions. For example, lipoproteins in the blood plasma carry lipids throughout the body. Other examples include catecholamine transport proteins that are targets for certain drugs. Nutrient and storage proteins are used to store essential nutrients. Seeds, for instance, store proteins for germinating seedlings. Ovalbumin in egg white and casein in milk are other common examples. Contractile or motile proteins enable cells and organisms to contract, change shape, or move. Tubulin, for example, is the protein that forms microtubules. Structural proteins provide strength and protection to biological structures, acting as supporting filaments, cables, or sheets. Collagen is a major component of tendons and cartilage, while elastin is found in ligaments. Defense proteins protect organisms from invaders. Immunoglobulins, or antibodies, recognize and neutralize bacteria. Fibrinogen and thrombin are defense proteins involved in blood clotting. Regulatory proteins help control cellular and physiological activities. G proteins, for instance, mediate the cellular response to many hormonal signals. Amino acids are the building blocks of proteins. They share a fundamental structure: a central carbon bonded to an amino group, a carboxyl group, and a hydrogen atom. This diagram illustrates the core structure of an amino acid, including the central carbon, the amino group, the carboxyl group, and the variable R group. There are 20 different types of amino acids used to make proteins. They all share the same essential structure but differ in their side groups, also known as the R groups. Amino acids link together via a covalent bond called a peptide bond. Polypeptides are polymers of amino acids connected by these bonds, forming long chains. This scene shows how amino acids link together to form a polypeptide chain. A peptide bond forms through dehydration synthesis, where a water molecule is removed as the carboxyl group of one amino acid joins the amino group of another. The resulting molecule is a polypeptide. Proteins are very large molecules. Some consist of a single polypeptide chain, while multi-subunit proteins have two or more. If some subunits are identical, the protein is called an oligomer. Proteins are organized into 4 levels: primary, secondary, tertiary, and quaternary structure. We will explore how amino acid sequence dictates protein function and structure. This image illustrates the 4 levels of protein organization. The primary structure is the sequence of amino acids linked by covalent peptide bonds, including any disulfide bonds. This sequence defines the protein's identity. Secondary structure refers to regular, recurring arrangements of adjacent amino acid residues in space. The most common types are the alpha helix and the beta conformation. Tertiary structure is the complete 3-dimensional shape of a single polypeptide chain. It describes the spatial relationship among all amino acids in that polypeptide. Quaternary structure applies to proteins with multiple polypeptide chains. It describes the spatial relationship of these subunits within the overall protein complex. The primary structure determines the final folded structure. Let's review the protein song to help remember these concepts. For independent practice, visit the provided link to explore and make note of the different types of amino acids used in making proteins. These are the references used in creating this video.